the really good thing is I've just realized I'm not in the lab, which means I can drink coffee from an open cup. Alright, the last video was for those students who are using continuous data and just collecting continuous data. But a number of students will be using discrete data. So either both the IV and the DV are discrete, or one is discrete. So I'm taking a scenario where I've decided that I'm going to compare the effect of 3 different types of sugars on the increase in the population of yeast cells. I'm going to pop that up here because that's my very rough research question. It's not a good research question, but this video is not about research questions. So let's go. Effect of different types of sugars. And to list them, I've decided to use glucose, which is what we used in our first experiment. I thought maybe yeast would like some fructose, and lactose, which is the sugar that you find in milk, on the increase in the number of yeast cells incubated over 24 hours. There we go, a rough research question. So I've got 3 things here. I've got glucose, I've got fructose, and I've got lactose. Those are the things that I've chosen to change, so those are my independent variable, and they are discrete. The number of yeast cells is also discrete. Those are the things that I'm going to be measuring. So I'm going to be counting yeast cells, and I'm not counting half yeast cells. I'm going to be whole yeast cells. So I've got a discrete independent variable, and I've got a discrete dependent variable. Now that means that I'm going to have to eventually do a column graph. And I'm going to need to know the mean for that, and then I'm going to have some form of determining whether the spread is such that it makes it statistically significant or insignificant. So I'm going to be using the standard error to determine that. All the generation of the graph, generation of the mean, generation of the standard error, putting it on the graph, those will all come in a later video. Remember that we are just setting up these spreadsheets so that we can enter our data. So for this one, it's slightly different to the continuous data one in that we want to have our dependent variables across the top here. So I'm going to put increase in number of yeast cells in glucose broth. First one, oh my goodness, my fingers are moving faster than, my brain's moving faster than my fingers. And as I said, I like to wrap the text so that it's neat. It looks a bit bulky to me, so I'm going to make it like that. And I really don't like it being at the bottom, so I'm going to pop it back up at the top, and that looks pretty to me. Alright, now basically I want to do the same, but I need a column for fructose and lactose. Not a fan of typing too much, so I'm going to copy that, paste it, and I'm going to shift these over a little bit, and I need to change this to fructose. I need to change this. Oh, changed the wrong one. I need to change this one to fructose. And this one to lactose. And there we have it, my 3 discrete independent variable parts. Now, being a good student, I've decided that I need to have 5 samples. I seem to be picking on the number 5. No reason for that, except that I just don't want to
try and do 10 because it's going to take too long. So I'm going to put that, so the number of repeats, that's going to go down this column here. So repeat number 1, 2, 3, 4, 5. There we go. So those are my repeats. I know that I'm going to be finding the mean in there. I know that I'm going to be finding the standard error. So I'm going to put that in there so that it's all ready to go. Now, just as before, I want to have a pretty table, not too fancy, just enough so that it looks neat. So I'm going to put a border, inside lines, and that table is nearly ready to go. It needs a heading. Really going to cheat here. I'm going to say table 1, still table 1 because it's a different experiment, and I am going to copy that. And there's my table heading. So this is now ready, once I've put the data in, to generate a graph, and it's ready to be copied and put straight into my report. We need to just think a little bit about this because we can't actually enter these data straight into this table. It says an increase in the number of cells in the glucose broth, but we're going to be counting them before and after. So it might be an idea to open another spreadsheet and to just do a table where you can enter your counts, number of cells before, number of cells after, and then use Excel to calculate the difference. If you need help with that, talk to one of your teachers because we'll be able to help you with that. Okay, so that's that one, ready to go. Now the last scenario we have is where we have discrete data, but we are comparing two things, and we know that we're going to be heading towards a t-test. For my student experiment, I've chosen to compare the number of damselfly larvae found in still water as opposed to fast flowing water. In emerald lakes. So I've got two things. I've got fast flowing water and I've got still water. So that's my IV. That's the thing that I'm choosing to change. The thing that I'm counting, the results that I'm getting are the number of damselfly larvae. They're also going to change depending on whether they come from fast flowing water or still water. I'm not choosing those. I'm choosing fast flowing water or still water. And again, we have to set this up so that we can easily generate a t-test from it and possibly a graph. So we're going to set it up in the same way that we set up those ones. So we're going to have two things. We've got number of larvae in still water and we've got the number of larvae in fast flowing. This time I've decided that I'm going to take samples from 8 different sites. I want to be sure that I have a large enough sample size. So I'm going to do 8. 1, 2, 3, 4, 5, 6, 7, 8. And again, I might want to do the mean. And I'm probably not going to do the standard error because I'm going to do the t-test. So I'm going to leave the standard error out just this once. Make my table, number, border, outline, inside, border done. Still haven't got a heading. Table 1. Copy that off there. And I'm going to put comparison of the number of damselfly larvae found in still water as opposed to fast flowing water in emerald lakes. There we go, good to go. And that is ready to take it further later on. Now this one, you're just doing a count, so you can just enter your results straight in there. You can either calculate the mean with an equation, or you could use the data analysis toolpack, which you're going to use anyway when you do the t-test. And again, that will be a separate video. Well, I hope you found that helpful. Please feel free to ask questions. We're here to help you.